السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. Second lecture in cytology. Today we are going to talk about mitochondria. Mitochondria is a membranous organelle containing enzymes responsible for cell respiration and energy production, and this is considered the powerhouse or the battery of the cell. This is a diagrammatic picture of a mitochondria. Mitochondria, the main function is production of energy. It contains outer membrane and inner membrane. The inner membrane form folds called cristae to increase the surface area. The number of the mitochondria is variable according to the cell which is present in. In highly active cell as liver cell, heart and muscle, the number of mitochondria may reach up to 1000. By LM, the mitochondria, when it is abandoned, it is responsible for the acidophilia of the cell. If you want to see the mitochondria well, you should stain it with iron hematoxylin and Janus green stains. This is an EM picture of the mitochondria, in which it's formed of two membranes, outer smooth membrane and inner membrane forming folds called cristae, and this is the matrix of the mitochondria. The outer membrane is smooth and contains protein called porins. These are permeable. The porins are proteins that cross a cell membrane, act as a pore in which the molecules can diffuse. It regulates the mitochondrial ATP and the calcium flux. While the inner membrane is less permeable, it is selective, project-forming shelves called cristae. The number corresponds to the energy needs. The more the cristae, the more the energy production, as these cristae increase the surface area containing the elementary particles, the global structure, which contains the enzymes, the respiratory enzymes, which is called oxidative phosphorylation enzyme. I make an oxidation of glucose, producing energy, and this energy will be stored in the form of ATP. The matrix. This is the outer membrane, and this is the inner, and this is the matrix. The matrix contains what? The oxidative enzymes of citric acid cycle or Krebs cycle, circular DNA, as the mitochondria can replicate itself. It contains all types of RNA messenger transfer and RNA. It contains electron-dense granules of calcium which act as a catalyst regulating the activity of the mitochondrial enzymes. This is an EM picture of the mitochondria formed of outer smooth membrane, inner membrane forming cristae, and this is the matrix containing calcium, DNA, and respiratory enzymes and RNA. What is the function of the mitochondria? Cell respiration and production of ATP, and it is the powerhouse of the cell. 50% of the energy is stored as ATP, and this is when needed. This ATP would change into ADP and producing energy, and 50% of the energy used to maintain a normal body temperature. They can form their protein by themselves, and they can undergo self-replication. A medical application of the mitochondria. Mitochondrial inheritance is usually maternal. The mutation of mitochondrial DNA results in muscle dysfunction. First, dear students, why the mitochondrial inheritance is maternal, as mitochondria present in the middle piece of the sperm, and fertilization occurs between the head of the sperm and the ova. So the middle piece doesn't enter in the fertilization, so no mitochondria will be transported from the father, just from the mother. This mitochondria is needed for energy production. So patients suffering from mutation in the DNA mitochondria will result in muscle weakness or dysfunction. Mention what is the self-replicating organelle. We have the mitochondria. The other organelle we're going to study is the endoplasmic reticulum. We have two types of endoplasmic reticulum. First of all, what is the meaning of an endoplasmic reticulum? It's the system of communicated membrane-bound tubules or vesicles, okay, called cisternae. We have two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. First, we're going to talk about the rough endoplasmic reticulum. Where I can find it? In a protein-producing cell. By LM, it appears basophilic. Why it is basophilic? As we said before in the first lecture, that anything acid in nature like to be stained with a basic stain, and they're called basophilic, and this basic stain is the hematoxylin and appears blue. By EM, it is a membrane-bound sheet, flattened tubules or cisternae intercommunicating together. At the outer surface, there is ribosomes that present on specific receptors called ribofurans. The ribosome present on the ribofuran by the large subunit of the ribosome. Here is a part of a cisternae of rough endoplasmic reticulum, and here are the ribosomes formed of small subunit and large subunit. Only the small and the large subunit fuse together only during protein synthesis. Okay, and this is the messenger RNA that took a copy from the DNA and passes from the nuclear pore to the cytoplasm. And this is the ribosome, which is formed of small subunits and large subunits. The large subunit sit on the rough endoplasmic reticulum cisternae at a receptor known as ribofuran by the large subunit. This is an EM picture of rough endoplasmic reticulum, appears as flattened, regular cisternae. In the outer surface, there are ribosomes. Again, EM picture of rough endoplasmic reticulum. Here is a protein-producing cell from where I know that. Inshallah, and I'm going to give you the nucleus in the last lecture. But here this nucleus is active, and I'm going to say why. Because it contains extended chromatin. It contains rough endoplasmic reticulum extending from the outer nuclear membrane to the cell membrane. This is a highly protein-producing cells, and this is mitochondria. This is a protein-producing cells containing mitochondria and rough endoplasmic reticulum. Very well formed rough endoplasmic reticulum. This cisternae extends from the nuclear membrane to the cell membrane. The functions of the rough endoplasmic reticulum. It accumulates the protein that formed by the ribosome. The rough endoplasmic reticulum does not produce proteins. It receives the protein that formed by the ribosome. It segregates the protein formed, makes something called initial glycosylation, starting to produce, to add glucose, packing the formed protein and bed off from the rough endoplasmic reticulum in a form of transfer vesicle. This transfer vesicle go to the Golgi apparatus for further secretion. The rough endoplasmic reticulum protects the cytoplasm from the hydrolytic enzymes produced inside the rough endoplasmic reticulum by the ribosomes. It may contain hydrolytic enzymes. If I do not put these hydrolytic enzymes in the tubules, the cytoplasm will be destroyed. One of the functions is intracellular transport, as we said, that extend from the nuclear membrane to the cell membrane. Okay, this photo is a diagram showing the nucleus, showing the rough endoplasmic reticulum, Golgi apparatus, and lysosome. Here, I just want to tell you the relation between the rough endoplasmic reticulum and the Golgi apparatus. The transfer vesicle arises from the rough endoplasmic reticulum entering to the Golgi apparatus. The other organelle is smooth endoplasmic reticulum. It appears as sacules that smooth, does not contain ribosomes on the surface. They are irregularly anastomosing. Okay. Branching and anastomosing tubules, and they are smooth and they does not contain ribosomes. Okay, and they are anastomosing in irregular network manner. The smooth endoplasmic reticulum present in steroid-secreting cells or lipid or fat steroid-secreting hormones forming cells as liver and endocrine glands. By LM, the smooth endoplasmic reticulum does not appear, but when abundant, it appears acidophilic. By EM, it appears as a membrane-bound tubules or vesicles with no ribosomes present on the surface. Here is EM picture of the smooth endoplasmic reticulum. You can see that it is irregular tubules as they anastomose irregularly, and there's no ribosomes on the surface. Here are EM picture of rough endoplasmic reticulum and smooth endoplasmic reticulum. What are the functions of the smooth endoplasmic reticulum? It produces phospholipids, it produces steroid hormones, help in muscle contraction because the smooth endoplasmic reticulum containing calcium inside it. This calcium it pumped from the smooth endoplasmic reticulum, allowing gliding of actin over myosin. One of the functions of the smooth endoplasmic reticulum is storage and breakdown of glycogen in the liver and in the muscle. Detoxification of drugs and hormones. So patients suffering from liver disease should avoid excess use of drugs, and as well as intracellular transport. Here is the calcium that pumps from the smooth endoplasmic reticulum that help in movement of myosin over actin. This is what's called Z line, and this is actin, and this is the myosin taking the actin inwards, making contraction. The organelle that predominate in steroid-secreting cell is the smooth endoplasmic reticulum. We are going to take organelle known as Golgi apparatus. Golgi is the scientist who discovered the apparatus. Okay, it is the organelle responsible for secretion. It is well developed in secretory cell. Okay, this is a diagrammatic, both of them diagrammatic picture. Here this diagrammatic picture for a cell. As you see, this cell is deeply basophilic, and you can see that there is an area in which I can see it pale. Deep basophilic means that it is a highly protein producing cell and appears pale due to presence of something that is not stained with hematoxylin and eosin, which is the Golgi apparatus. So by hematoxylin and eosin, the Golgi apparatus will not appear, and so this area is called negative Golgi image. Okay, here again is LN picture of negative Golgi image. In plasma cell, which is a protein producing cell forming immunoglobulins, negative Golgi image is evident. We can see the negative Golgi image when stained with hematoxylin and eosin. If you want to stain the Golgi apparatus, stain it with silver. It appears brown granules or fibrils. Where I can find the Golgi apparatus, it is present at the apical region between the nucleus and the secretory pore in secretory cell, and present around the nucleus perinuclear in the nerve cell. And a picture of Golgi apparatus in secretory cell stained by silver. These are cells close to each other, resting on a basement membrane, and this is the nucleus, and this is the Golgi apparatus. It's present at the apex between the apical surface and the nuclei. Why it's present apical? As the secretory vesicle move a small distance and open by exocytosis to the lumen. Here is the Golgi apparatus perinuclear in the nerve cell and stained by silver. The organelle that stained as the brown fibrils with silver is Golgi apparatus. Here is a diagrammatic picture showing the relation between the rough endoplasmic reticulum and the Golgi apparatus. The secreted structure that the protein passes from the rough endoplasmic reticulum in the form of transfer vesicle. This transfer vesicle go to the Golgi apparatus. Changes occurs in the Golgi apparatus, and a secretory product arises from the other face of the Golgi apparatus. This is the structure of the Golgi apparatus. As we see, intercommunicating tubules, those two surfaces, cis face or immature face or convex face, and trans face or concave or mature. To the cis face, transfer vesicle moves towards it. The cis face receives transfer vesicle, while the trans face or the mature face release secretory vesicle or lysosome. The EM picture of Golgi apparatus: stacked group saucer-shaped flattened vesicles. They are flattened membrane bound. They have narrow lumen and expanded ends, contain low electron dense material. They are 3 to 10 parallel saccules arranged one above the other, forming stack. They are interconnected together, and they have two faces: the convex or deforming face that receive the transfer vesicle, while the concave or mature face from which the vesicles bud off. This vesicle is either secretory vesicle or lysosome. This is the stack of the Golgi apparatus. Here two Golgi apparatus showing the stack of vesicles. Okay, stack of saccules, and here is intercommunicating together. The secretory vesicle moves to the cell membrane, discharging this content by exocytosis. The Golgi apparatus also produces lysosomes containing hydrolytic enzymes. What is the function of the Golgi apparatus? It concentrates and accumulates products received from the rough endoplasmic reticulum. Making chemical modification of the protein by adding carbohydrates or sulfates, producing secretory vesicles which may be hormones, formation of lysosomes, and maintenance of the cell membrane. How the Golgi apparatus maintain the cell membrane? Here is a diagrammatic picture showing the cell membrane, and these are the secretory vesicles containing contents that will be released by exocytosis. The vesicle fuses with the cell membrane, then the content moves out the cell while the coat that surrounds the vesicle will be added to the cell membrane. That's why it's maintain the integrity of the cell membrane.